Hello, my name is Matt Solba. I'm an assistant professor in the Department of Medicine, Division of Infectious Diseases. It is a pleasure to meet you virtually, and I'm very happy to tell you a little bit more about the work we do in the lab. So I'm going to share some slides with you. The first slide is just the landing page for our website. And so welcome to the Solba Lab. We're a microbiology lab. We're housed within the Division of Infectious Diseases at the University of Pittsburgh School of Medicine. And we're located on the 8th floor of the SCAPE building. And so the primary mission of our lab is to promote health and happiness through scientific discovery, through a research and educational mission. Our research focuses on the molecular mechanisms of bacterial mutagenesis and adaptation. We have a real focus on antibiotic resistance. As you know, antibiotic resistance poses a severe public health threat, not just here in the United States, but around the globe. In the 1940s, human beings started mass producing antibiotics and using them to treat infections very successfully. And in fact, it's the primary reason why we're enjoying the modern era of medicine, as it were. Antibiotics are critical to treating a whole array of common diseases. However, bacteria have a remarkable ability to adapt to antibiotics. And unfortunately, the pace of antibiotic resistance is starting to be faster than our ability to make new antibiotics to counteract resistance. And this is very scary. And so what we focus on in the lab is just trying to understand what are the molecular mechanisms that underpin this remarkable ability that bacteria have. How is it that they can do this so rapidly? Really, the center of our thinking in the lab is around mutagenesis. Mutation and genetic diversity are the substrate upon which evolution acts. And you can learn a lot by looking at mutations. So, for example, some bacteria, when encountering antibiotic stress or other stressful situations in the environment, they upregulate their mutation rates, and that can hasten the acquisition of antibiotic resistance. The other way that you can learn stuff from looking at mutations is by tracking mutations in the bacterial population that's under stress over time. And by doing that, you can learn what genes are under positive selection, which can clue you in as to what pathways are really important for the bacteria to evade antibiotics and acquire things like tolerance phenotypes. And so we use both approaches in the lab. Our research is really focused on two genetic pathways. Now, the first one is the DNA damage response, or the SOS response. We use E. coli as a model organism in this case. The job of the SOS response is to counteract DNA damage. So in the setting of a DNA damaging event, which can actually happen with many of the antibiotics that we use, there's a series of about 40 genes that are upregulated. And the job of those genes is really to counteract the DNA damage, to go find it, cut it out, or repair it, and allow the cell to go on its merry way so it can survive that insult. However, there's a couple of genes in the pathway which encode highly pro-mutagenic polymerases. And this can up the mutation rate and hasten the acquisition of resistance. And so our research in the lab is trying to understand how these 40 genes are regulated, the timing of their regulation, and also what the mutagenic consequences of these air-prone polymerases are. The other pathway that we're looking at is the stringent response pathway. And here we're actually using clinical isolates of Staph aureus. We got into this project by tracking evolution in bloodstream infections in patients in the hospital. We found a big positive selection signal in this gene called REL. And so we got interested in this pathway. And we're currently studying purifying the REL enzyme and studying its activity in vitro to try to understand this mechanism. The way that the stringent response works is that in the setting of cellular stress, particularly amino acid deprivation, tRNAs become uncharged, meaning that their cognate amino acids are no longer available to covalently link to the tRNA. When these uncharged tRNAs bind to the ribosome, this REL enzyme can sense that. And it turns on its synthase activity, which essentially produces this polyphosphorylated guanine analog called PPPGPP. This alarm molecule has a lot of downstream consequences and can changed global gene regulation and essentially turned cells into these little tanks that have very slow metabolism and slow growth rates and allow them to develop a multi-drug antibiotic tolerance phenotype. And so we're interested in exactly how REL is regulating its activities with the hope that we can discover a way to intervene on this pathway as well. So I hope that brief overview gives you an idea of what we're doing in the lab. If you'd like to learn more, please feel free to reach out. I'm going to leave you with my contact information here on the last slide. And there's my email address right there. And so please shoot me an email if you'd like to talk further. And thanks so much for your time and attention.